Hello guys, welcome to IB360. Today we will start with cell membrane of eukaryotic cell. In the previous video we had talked about cell wall, and I had mentioned that we will discuss eukaryotic cell broadly on the basis of these four topics: cell wall, plasma membrane, cytoplasm and nucleus. So today we will discuss plasma membrane of eukaryotic cell. Okay, so in 1838, Schwann, the German zoologist, he discovered plasma membrane, and after that, Nageli and Kramer, they coined the term plasma membrane. So the thing was discovered by Schwann, who is one of the proponents of the cell theory, and Nageli and Kramer in 1855 they coined the term plasma membrane. Much later, in 1931, Plowey he renamed the thing as plasma lemma. But originally, the term cell membrane that was coined by Nageli and Kramer. So membranes, not only they are compartmentalizing the cell from the environment, but the membranes are also occurring inside the cytoplasm of eukaryotic cells, as they cover several cell organelles like nucleus, mitochondria, plastids, lysosomes, Golgi bodies, peroxisomes, etc. So and we see vacuoles, they are also separated from the cytoplasm by a membrane called tonoplast. So all membranes, whether external or internal, are now called cell membranes or biomembranes. And these words are very important when we describe cell membrane: quasi-fluid, elastic, pliable, dynamic and film-like. Quasi-fluid means it is partially fluid, it is not entirely solid, it is not entirely fluid. Elastic means even if we are stretching it, it will return to its original conformation. And pliable means flexible. Dynamic means it is always under some sort of motion. It is not exactly stable. And by this word, they are actually implying the lateral movements of various components, which we will later see present inside cell membrane of eukaryotic cell. And film, like how in this diagram we can see that like an oil film over water. Based on these words, several MCQs are often seen to come. We will see one of them. So the question here is, plasma lemma can be described using words: pliable, selectively permeable for water, quasi-fluid, or both A and C. So we have already seen these terms, pliable and quasi-fluid, but someone might wonder that why is this not the answer? Obviously you can understand the answer is D, both A and C, but why not B? Because the cell membrane is not selectively permeable for water, rather it is semi-permeable for water. And there is a significant difference between the word selectively permeable and semi-permeable. Semi-permeable for water and selectively permeable for solutes. Okay, if I draw a schematic diagram of the cell membrane, suppose, so water molecules are small enough to penetrate these holes or these pores, and this phenomena is semi-permeability. But for selective permeability, size is not a factor. Size is not a factor. Here the membrane chooses the molecules that it will allow to pass through it, and this is mainly performed via various proteins which are embedded into the membrane. These are channel proteins or transmembrane proteins or carrier proteins. So when we will study bulk transport in the later classes, we will deal with this topic in much more details, that is selective permeability of plasma membrane. But for now you should remember that the plasma membrane is selectively permeable for solutes, because solutes are large enough and they cannot penetrate through these small pores. On the other hand, water molecules they can. And so what for water, the cell membrane is semi-permeable and not selectively permeable. So this word is wrong, and that is why option B is incorrect. Our answers would be pliable and quasi-fluid. Both we have seen. Pliable because it is obviously flexible, and quasi-fluid because it is partially solid and partially fluid, not entirely either of them. Okay. Now, composition of cell wall. So these values which we see, these actually varies from cell to cell, and that is why we have shown in the form of ranges. Lipids, they occupy 79% actually, to be more precise. Proteins 20 to 70%, carbohydrates 1 to 5%, and water 20%. And the ratio of protein and lipid, protein is to lipid ratio, this is actually not constant, and it varies in different membranes. For example, human erythrocyte membrane, mane RBC membrane, contains 52% protein and 40% lipid, while myelinated neurons, they have myelinated neurons 20% protein and 80% lipid. That is why these ranges are shown, because they are not constant, they varies from cell to cell. The important lipids of the membrane, they are phosphoglycerides or phospholipids. Phospholipids are some 100 types, so that also we can mention. Phospholipids are some 100 types, almost, not exactly 100, but almost some 100 types. They can also be called phosphoglycerides. And carbohydrates, they are present in the membrane as branched or unbranched oligosaccharides, for example hexose, hexoamine, sialic acid, etc. And as far as proteins are concerned, they can be fibrous or globular, they can be structural, they can be carrier, they can be receptor, they can be enzymatic. Structurally, proteins are either completely embedded inside or they can be extrinsic. That we will talk about in the later part of this video. Okay, and water is 20%. Another important thing which we should know now is the word amphipathic. Amphipathic. So, by amphipathic, it means that the particular molecule will possess both hydrophilic and hydrophobic ends. Hydrophilic means water loving, and hydrophobic is water repelling. Amphipathic molecules are both lipids as well as proteins. Lipids, they have the hydrophilic region in the form of a head, which is projecting outward towards the ECF, extracellular fluid, while the hydrophobic part contains two tails of fatty acids, and they're usually projecting towards the center of the membrane, towards the inner part of the membrane or core of the membrane. And this results in the formation of a lipid bilayer. So it is somewhat like this. So this is the outer surface, and this is the inner surface of the lipid bilayer, and these heads are hydrophilic, which are projecting towards the ECF. This is projecting towards the cytosol, and this in the middle, which is projecting towards the core or inner environment of the cell membrane, these are the hydrophobic chains of fatty acids. So this feature of biomembranes, they are aptly described by the word amphipathic. Amphipathic nature is also shown by proteins. Protein molecules also possess both polar and non-polar side chains. Usually their polar hydrophobic linkages are towards the outer side, and the non-polar or hydrophobic linkages are either kept folded inside or used to establish connections with hydrophobic part of the lipids. So that is why with both lipids and, to be more precise, with transmembrane proteins, we can use this word amphipathic. Okay. Now the various models of biomembrane. Now several types of models have been put forward to explain the structure of a biomembrane. The most accepted one is Mosaic model. However, before Mosaic model, several lamellar models have been proposed, and these lamellar models are also known as sandwich models. And these two are almost similar: Danieli, Davison model and Robertson model. The first lamellar model was proposed by James Danieli and Hugh Davison in 1935 on the basis of their physiological studies. And according to Danieli and Davison, a biomembrane contains four molecular layers, which are something like this. So these are the bilipid layers, and above these bilipid layers there would be layers of globular proteins, both towards cytosol and both to ECF. So these would be globular proteins, and these would be lipid bilayer. And another interesting thing is they said that 100% composition is it's almost like a sandwich. This is 100% globular protein, this is 100% lipid bilayer, and this is again 100% globular protein. This was the thing which Danieli-Davison model propounded. Similar thing was propounded by Robertson model, but what he said was slightly different in terms of the nature of this protein. They said that these extended proteins are actually beta protein molecules, so not much difference as such when we look over the general idea. But the fluid mosaic model, which is mainly taught in our syllabus, that says that the membrane does not have a uniform disposition of lipids and proteins. So this is not 100, rather uh, is a mosaic of the two. So there is no uniform disposition of lipids and proteins. We will see more about this fluid mosaic model, which is the most recent model of a biomembrane proposed by Sanger and Nicholson in 1972. And this is the most practical one also, because as per the lamellar models, the biomembranes are supposed to be stable, layered, very properly delineated from each other, very uniform, like 100% globular proteins, 100% bilayer of lipids, again 100% globular protein, according to the lamellar model proposes. This was a scenario. So according to fluid mosaic model, there is the constancy is not always maintained in the composition. And as from the very beginning I was telling that we have various composition of lipids and proteins, like lipids are either 20 to 80%, or proteins are 20 to 70%. But here they will say this region is 100% protein and this region is 100% bilayer of phospholipids. So this was the Davison-Danieli model, and this is the fluid mosaic model proposed by Sanger and Nicholson in 1972. Now another important thing which I was talking about is the quasi-fluid nature of the fluid mosaic model. See, these are the proteins, and proteins, if we differentiate proteins structurally proteins can be structural differentiation, they can be either intrinsic or extrinsic. Intrinsic protein, this is one example of intrinsic protein, embedded proteins as they are saying, completely embedded within the bilayer. And the part of protein which is forming bond with the bilayer is hydrophobic, and the externally protruding part is hydrophilic. And these are also called transmembrane proteins, and they can participate in various jobs, like they can act as carrier, they can act as channels. Carrier proteins and channel proteins, the job is not same. They can also act as receptors for hormones, they can also act as enzymes sometimes. So integral or intrinsic protein, they are 70% of the total membrane proteins, and they cannot be extracted without disrupting the entire cell membrane, like in case of detergents that they do. And the integral proteins pass into the lipid bilayer to different depths, and so they are also called tunnel proteins. Various names are given. And some helical structures may extend beyond the two surfaces, and these are carbohydrates they're mentioning. To be more precise, these are glycophorins. Okay, then there are also extrinsic proteins. Extrinsic proteins, they are entirely protruding out of the cell, and they are located superficially on the two surfaces, on the external face. For example, spectrin. Spectrin is an extrinsic protein example. The extrinsic proteins are attached covalently to phospholipid head. Anchored proteins they're called, or non-covalently to transmembrane proteins, and the latter can be separated with mild treatment only. The proteins overall provide structural and functional specificity to the membranes. Further, since the lipid bilayer is quasi-fluid, the membrane proteins may shift laterally also. As I was talking at the very beginning, their flexibility and dynamism that they impart to the membrane. These lateral movement, they can laterally move, they can also flex like this. They can rotate laterally as well as vertically. And this imparts the dynamism to the membrane. So the membrane is not at all stable, or not at all delineated into 100% of globular protein, 100% of lipid bilayer. Rather, it is more like a mosaic. So the term fluid mosaic model is aptly used. Okay, now asymmetry of biomembranes. Lipids present in the outer and inner side of the bilayer are commonly different. So lecithin. Lecithin on the outer side and cephalin on the inner side of RBC membrane is a very famous example for this. Lecithin on outer surface. By outer surface, I mean if this is suppose a schematic diagram of a part of the cell membrane. So this is facing the ECF, and this one is facing the cytosol. So this one is the outer surface and this one is the inner surface. So on the outer surface, the lipids which are attached, they are lecithin. These lipids are lecithin, and on the inner surface, cephalin. And this kind of pattern is seen in RBCs. So this is a very famous example for asymmetry in biomembranes. Amount of extrinsic proteins differ on the two sides. They are more abundant on the side, on the inner surface, than on the outer surface. Here the extrinsic proteins are more abundant than compared to on the outer surface. Oligosaccharides attached to external surfaces of lipids and proteins are absent on the inner surface. Yes, this is one more important asymmetrical feature of biomembrane. So today we have almost done with cell membrane. We have talked about the composition, we have talked about the various models which are proposed, the most accepted and most practical being the mosaic model, the fluid mosaic model proposed by Sanger and Nicholson in 1972. We have talked about the particular model. There are this bilayer of phospholipid with varying degree of proteins embedded inside or projecting outward. So they may be intrinsic or extrinsic proteins. Examples we have studied, and their functions we have also looked, and what is the essential difference between the fluid mosaic model and these Daniel Davison model, that also we have seen. And then we have talked about asymmetry of biomolecules. So for today we are ending our class here. Hope you enjoy the video. Please do like, share and subscribe our channel IB360, and stay safe. Thank you.